All right, welcome to podcast number, I don't know, 43, I think. We've been talking about a lot of evolution. This will be our last podcast. We've talked about a whole lot about the details of how these small microevolutionary changes occur, how these small changes in the frequencies of alleles build up over time. But what is it that actually creates a new species? What is it that leads to a new species arising? How do we know when that's even happened? So the origin of species. We are in this podcast going to look at those macroevolutionary changes. How do we get from a dinosaur to a bird, or from some dog-like thing to a whale? How do we get new species? So the word for new species, creation of species, is speciation, and that is definitely a discussion of macroevolution. But remember, macroevolution is really just lots of microevolution that has accumulated over time. It's all very tiny increments. It's just that as we would look at things, we'd say, wow, that thing's really different from what we started with. And where along that line do we decide that a new species has arisen? Okay, so we're looking, this is kind of an important phrase, lots of accumulated change, not just at one gene locus, but at multiple gene loci, where we've had not just maybe fur color, but behavioral changes, all kinds of stuff that lead to a new species. Very quickly, here's a couple of words that I honestly don't know that you'll ever see, but just in case, I don't want this to be the year that you see them and I didn't teach them to you. Two main patterns we see in speciation. One is where one species simply changes into something else over time. That is referred to as anagenesis, okay? In which case, this species here would no longer exist, that would transform into this new species. Cladogenesis, on the other hand, is speciation in which the parent species would continue to exist, but a new species would branch off from that. So obviously this one creates diversity, lots of species existing at the same time, whereas this one would not increase diversity so much. Okay? So if we're going to talk about creating species, it's really helpful to have a definition of what a species is. What is it? And you need to be aware that there's lots and lots of definitions of species out there. And we just need to find one that works. And they don't necessarily all work for all situations. So it's a little bit of a gray area. When you are a student learning, you like to be, just teach me the right and the wrong. And unfortunately, there's a lot of gray that we don't like to teach and we don't like to learn all the time. But the concept that we most often rely on is this one that was put forth by Ernst Mayr many years ago, called the biological species concept. And basically, by this definition, a species is a population of animals whose members can interbreed with each other, and when they do interbreed, they can produce viable, that means offspring that can live, and fertile, they can live and reproduce offspring, fertile offspring. Okay? All right, so that is a workable definition. However, if you think about it, I mean, look at all these different people. They look really, really different. Yet all of these people are one species because, I mean, they meet this definition. They can interbreed and produce viable, fertile offspring that can reproduce themselves. These two animals here, though, are different species. Evidently, I mean, look at them. They look identical. I couldn't tell them apart. Physically anyway, but they cannot interbreed or produce viable fertile offspring, or perhaps they just don't. It's kind of shades of gray here, but it's a tricky definition to get your hand around, and it's not just one definition that exists either. Okay, what is it that keeps them from interbreeding here? They have different songs, so that if this one sings, this one does not recognize that song, different behaviors, and that prevents them from interbreeding in nature. Could you force them to interbreed in the lab? Perhaps. An idea that is really central to this biological species concept that Ernst Mayr we just got is the idea of reproductive isolation. Let me go back. These guys are reproductively isolated from each other. These guys and gals are not. There is something that prevents them from interbreeding and producing fertile offspring. Okay, so in order to be separate, distinct species, first of all, you can't intermingle with other species. You're distinct. You're separate. And that means there must be barriers that prevent reproduction between you and organisms that are not of your species. So again, let's go back and look at these guys. These guys have some kind of barrier that separates them, prevents them from interbreeding. Perhaps it's their song, whatever. So if you are a distinct species, you and your other members of your species are distinct, you are isolated from members of all other species, and you can only reproduce with each other. Okay? Now, when we're talking about barriers to reproduction, we are not talking geographical barriers. They have to be biological, innate to the organisms, not just because I have two squirrels and I'm going to put one, at Monacan High School and one over in Los Angeles, California, and say, well, they're two species because they can't interbreed because they're too far apart. That doesn't count. True reproductive isolation means we are going to prevent the organisms from breeding even if their ranges overlap with each other. Okay? There are two main types of reproductive barriers. Again, remember, this is what makes a species is not being able to reproduce with others. So we've kind of got to get a handle on this before we can talk about creation of new species. So creation of these reproductive barriers gives us new species. And there are two main types of reproductive barriers: pre-zygotic, or before the zygote is formed, barriers, and post-zygotic, or after the zygote is formed, reproductive barriers. Okay? So pre-zygotic barriers prevent a zygote from ever forming, whereas post-zygotic barriers may allow the zygote to form. We may actually have sex, but the offspring will fail in some way. They're going to be weak and not work in some way. All right, so what we're going to do is look at some barriers to reproduction. The first ones we will look at are pre-zygotic. Ecological isolation is a pre-zygotic barrier. Okay? And what this says is that species may occur in the same area, right here in my yard, for example, but they occupy different habitats in my yard so that they just don't encounter each other. And if they don't encounter each other, they don't interbreed. Okay? They are reproductively isolated. This is a great example. There's a garter snake. There's a distinct species here and here. They live in the same general area, but one is aquatic, the other is terrestrial, and because of that, they will not interbreed. They don't interact with each other. Okay? Lions and tigers can hybridize. They can. You've all heard about ligers and all that. But they live in different habitats, and they do not interbreed in nature. They are ecologically isolated. So again, just because we can force someone to reproduce doesn't mean that they are not distinct species. Okay? So Think of it, here's two birds, very similar looking perhaps, but one always hangs out in the canopy, one hangs out in the understory. They're ecologically isolated. Okay? Behavioral isolation. Bird song. If you don't sing the right song, you're not going to mate. So, unique behavioral patterns will prevent zygotes from forming and isolate animals reproductively from each other. Blue-footed boobies have just elaborate dances that they have to do, and if there's a similar species but the dance is not correct, then they will not mate. They are reproductively isolated. It identifies members of the species. Okay? Reproductive isolation. Temporal isolation is another type of pre-zygotic barrier. We're not even going to let the animals make a zygote. Why? If you breed during different times of day, during different seasons, during whatever, then you won't interbreed. For example, these two types of skunk, eastern and western spotted skunks, they overlap in their range, but the eastern one up here that looks to me identical to the western one mates in late winter, and the western mates in late summer. They're not going to interbreed. They are reproductively isolated from each other. They are not mating at the same times of year, so no zygotes will be made between these two very, very similar looking species of skunk. That's isolation in time. Mechanical isolation. Basically, the parts don't work together here. You can imagine that I don't know, this is probably a bad example, but a Great Dane and a Chihuahua, the parts don't work. Reproductive isolation will occur if the parts cannot be made to fit together. Plants are a great way to look at this. Even in very closely related species, the flowers will often have very distinctly different appearances that attract different kinds of pollinators, and so cross-pollination does not occur. The flower shape can be distinctly different for different pollinators, for example, and this one can only pollinate this flower, this one can only pollinate that, and they do not cross-pollinate. So you end up with reproductive isolation. For many insects, male and female sex organs, if you look here, these are all damselfly reproductive organs, but they're all very, very different, and you don't get cross-fertilization between the species because the parts simply won't fit. Okay. Now, gametic isolation. Here, maybe reproduction, sexual activity occurs, but a zygote cannot be made because the sperm of one species simply cannot fertilize the eggs of another species. For example, if there are certain receptors on egg and sperm that fit together, if what you're seeing here is a receptor and an identification marker on the surface of sperm and egg here, and they fit. So this sperm could fertilize this egg. But if the sperm does not have the correct marker, it can't fit with the receptor, and so even though sperm and egg come really close, there's not going to be any fertilization. Okay? Now, post-zygotic reproductive barriers. Again, we're still going to prevent the creation of fertile offspring, but this time we actually get a zygote, but we're going to prevent that hybrid offspring from developing into a viable fertile adult. There are 3 ways to do that. We are going to make it harder for the hybrid to survive. We're going to make it harder for the hybrid to be fertile, and the hybrid will eventually just break down. So let's talk about that. So, in reduced hybrid fertility, you may get one species mating with another, and you may get an organism, and it may be very healthy, but it's sterile. It cannot reproduce organisms, offspring of its own. That is the classic donkey plus horse gives us a mule, and this animal's healthy, but it can't reproduce on its own. Okay, this often occurs when the chromosomes of the parents differ in number, and so they can come together and make the offspring, but the offspring can't do meiosis properly and can't make fertile sex cells. Okay, so there's your horse, your donkey, and your mule. They are vigorous, healthy, but sterile. Okay, and what you can see here, horses have 64 chromosomes, donkeys have 62, so the gamete here has 30, the gamete would have 32 chromosomes, this would have 31 chromosomes, and the mules would have 63, and that's an odd number, and that doesn't work well with meiosis. Okay? All right, reduced hybrid viability. This means that basically the organism that is created, the hybrid that is created, does not survive well. Most hybrids don't develop all the way, and those that do are frail and they don't reproduce. So in this case, the hybrid simply isn't viable. It doesn't survive. Okay? Hybrid breakdown is when two parent species interbreed and you get a hybrid, and it's viable and it's fertile at first in the first generation. But when those hybrids start having subsequent generations, the offspring don't work out well. They are feeble or they are sterile, so it's not really viable in the long run. This is a nice diagram. It's straight out of your textbook, but this shows you pre-zygotic reproductive barriers and post-zygotic reproductive barriers, and individuals of different species. If they fail at any point due to any one of these barriers, then they are distinct species. But if they make it through all these different barriers and raise viable fertile offspring, then these would not be considered different species anymore. They would be considered one species. Okay, so these are all barriers that would make two species be separate from each other. Okay? Excuse me. All right. Please know that this, again, biological species concept doesn't always work. It's lousy with fossils because you can't watch fossils breed. So basically you're stuck with just using morphology. What do they look like to categorize fossils? Asexual life forms don't have sex, so the biological species concept doesn't work with them either. Those are some bacteria there. And even some sexual life forms. Coyotes can interbreed with wolves and dogs, yet they are distinct species. And where do you draw the line? So it's not always clear cut. All right. So we've talked about what this idea of reproductive isolation means. So two species are distinct. They cannot reproduce with each other. But how do they get that way? How do new species originate? How do they get isolated? Okay? Now, the easiest way to do that is to simply geographically separate organisms. And you might say, but wait, I thought geography didn't matter. It doesn't, but if you have a group of organisms and they're all together geographically, here's my squirrels, and they're all in the same area, but I put a big barrier up between them. Are these guys over here genetically identical to these guys? Probably not. I've probably got a few different genes on this side of the barrier just by chance than I do on this side. And if I leave this barrier up for 1,000,000 years, it may be that I come back and these guys have just gone on a different evolutionary path than these guys, such that if I remove this barrier, they'd see each other and they just wouldn't reproduce because so many differences had accumulated among them. Okay? Isolated populations do take different evolutionary paths and will evolve independently of each other if they are separated geographically somehow. This is called, and you do need this term, they love this term, allopatric speciation, which is speciation that occurs because you geographically somehow separate species with some kind of barrier, and so, populations, and so this population, even though they may have started out being essentially the same species, this population will take a different evolutionary path than this one, okay? But another way to get a new species is a little less intuitive, and it's called sympatric speciation. So we got allopatric, is got your geographic separation as your starting point. In this case, you don't have a geographic separation. You have, let me get a better, this is talking back about allopatric again. I'll go back and give you an example. Sympatric speciation is essentially, let me find my good little diagram I like. I hope it's here. No, I must have replaced it, this one on the fishies. Let me go back. Okay. Suppose a mutation arises, something happens that causes a fish to give birth to a different kind of fish, and somehow this fish is so different from that fish that it cannot interbreed back with them. So these guys will interbreed with each other, and boom, you've got a new species. This happens a lot with plants because plants have wacky chromosomes and they can asexually reproduce. So if you've got a bunch of trees here, and you might get one tree from this group that due to a mutation pops up different, and it cannot interbreed back with these trees, but it can asexually reproduce itself, so it can start a whole new population within this same geographic area. That is sympatric speciation, okay? So allopatric involves geographic separation, sympatric involves genetic accidents, and often these are chromosomal. Here's a great example of allopatric speciation. There are two species of squirrel, chipmunk, whatever. If I saw this, I'd call it a chipmunk, in the Grand Canyon. One is on this side, one is on this side. They got separated by the Colorado River carving out the Grand Canyon, and they diverged. And if you put them together, they don't interbreed. Okay? Death Valley pupfish, another great example. Believe it or not, Death Valley was once not Death Valley. It was very interconnected lakes all around, and the drying began to happen around 10,000 years ago, which left just isolated springs here or there in the desert. And each spring is a little bit different. Each spring differs in its temperature and its salt content, and they're very small, just a few meters across. Okay? In these little isolated springs live little fish that are called pupfish, and each little different spring has its own species, and it's found nowhere else. It is thought that there was one ancestor that had the whole area of the Death Valley when it was a bunch of interconnected lakes, but when these lakes got separated, its descendants slowly diverged from each other and became specifically adapted to the little spring that they lived in. So once they were separated, the ancestral populations diverged and created all the different species found today. They were reproductively isolated from each other after being separated for so long. Again, this is allopatric speciation, okay? Geographic separation, even if it's just in different little pools of water. Okay? This is also a good example. The pupfish example is a good example of adaptive radiation, where one pupfish gave rise to many different ones because of the way evolution, natural selection occurred in each of the different pools. Okay? Darwin's finches is a good example. The different islands where geographic separation occurred, so the one ancestor gave rise to many. Okay, and then sympatric speciation. So how can sympatric, what kind of chromosomal weirdness can occur to allow a new species arise right there in the midst due to a genetic accident? Chromosomal mutations are great examples. A new species can be generated in one generation if there is a genetic change that prevents it from breeding back with its parent population. Polyploidy is a great example of this. An accident can occur during cell division that basically ends up, and often this is a plant, getting an extra set of chromosomes. That means it can't breed back with its parent population, but it can breed with itself asexually, and you can get a new population of plants. Okay? Polyploid just means many sets of chromosomes. Okay? I have seen these terms come up on tests. But let's just not. All right. Well, let me see if I can give you a very brief definition. Two types of multiple sets of chromosomes. Autoploidy are basically when you have one individual that arises with these multiple sets of chromosomes, but it's arised from a single species. Okay, that's auto. Auto means self. So one species somebody had a genetic accident, had an offspring that had multiple sets of chromosomes, and now it can't breed back with the parents, but it can reproduce asexually. Okay? Alloploidy is when you get a hybridization event. You've got a species here and a species here, and they do cross, and they've got a weird chromosome number so that the offspring can't breed back with the parents, but it can self-perpetuate itself if it's a plant. This is a great example. There is a species of salt marsh grass in England that actually is called Spartina, Spartina maritima. We have a Spartina species here, alterniflora. If you go to the bay, you will see it. It grows all over the place. They hybridized and have now made what is really a pest species of marsh grass in Europe. The American species hybridized with the European species, and they can look at the chromosomes and see that that's true. This is the European species. Its diploid number of chromosomes is 60. This is our species, and it's got 62, and the hybrid has got 122, which is exactly what you would expect. This hybrid can't breed back with either parent, but it can self-fertilize and breed with itself, and it's very vigorous and apparently has become a bit of a pest in Europe. This is the diagram I wanted. This is the one you should remember. Allopatric, sympatric. Allopatric, we get speciation because we geographically separate, and then these guys go off on a different evolutionary path over time than these guys. They can't interbreed, they're too far apart, whatever. In this case, we have these trees, and there's a genetic accident, and a different type of tree rises up right in the middle due to a chromosomal anomaly or whatever. They can't breed back with their parent plants because they're too different, so they asexually reproduce and interbreed with each other. Sympatric, allopatric. You need this slide. Okay? All right, we're going to stop there, and we'll catch you at the next podcast. Take care.